Hello, my name is Lilach Lerman. I'm a professor of medicine and physiology at the Mayo Clinic. I'm the associate chair for the Division of Nephrology and Hypertension at the Mayo Clinic, and I've been involved in translational hypertension research for over 20 years now. In particular, my research has focused on studying atherosclerotic renovascular disease, an important disease that afflicts almost 7% of adults over 65 years of age. Atherosclerotic renovascular disease can cause not only damage to the kidney, but can also cause development of hypertension and serious effects on the cardiovascular system, as well as on the contralateral non-stenotic kidney. Therefore, there is a very important motivation and rationalization to try to treat the post-stenotic kidney in order to try to attenuate these potential side effects. Over the years, we have investigated a number of interventional techniques to attempt and blunt renal injury in the post-stenotic kidney. These involve both revascularization of the stenosis, as well as delivery of specific targeted drugs or cell-based therapy in order to blunt renal injury. Mostly these approaches have been successful in attenuating kidney injury, although it appears that in order to attenuate hypertension, revascularization or other techniques of decreasing blood pressure are still required. These approaches can actually be translated into the clinical arena because at least two of our approaches and our strategies have been translated and have already instigated clinical trials to try to determine the effects of both cell-based therapy and a mitochondrial targeted peptide on the kidney in order to try and decrease renal injury in this disease. Many of our studies have been performed in animal models of renal artery stenosis that we induce using implantation of a local irritant coil in one renal artery, which leads to development of hypertension within about a week. We then feed the animals with a high cholesterol diet or a diet that leads to obesity so that we can study the effects of comorbidities on development of renal injury and of renovascular hypertension. This model also lends itself to the possibility of performing percutaneous transluminal renal angioplasty, PTRA, which is akin to balloon angioplasty in the coronary arteries and causes a fast decline in blood pressure. In order to study the post-stenotic kidney and the non-stenotic contralateral kidney, we have been employing sophisticated imaging technique such as multi-detector computed tomography or MDCT, as well as more recently magnetic resonance imaging, which we have been using in order to study renal blood flow and intrarenal oxygenation. These techniques can shed light on the occurrences and the pathophysiological evolution of kidney injury within the stenotic kidney, as well as within the contralateral kidney, and at the same time also studying the effects of hypertension on the heart. Furthermore, these techniques can be translated to humans because these machines are also available clinically. Over the course of our studies, we have found that the mechanisms of kidney injury in this disease involve increased oxidative stress, inflammation, apoptosis, and development of fibrosis in the stenotic kidney. Furthermore, we found a significant loss in the number of microvessels in the post-stenotic kidney. All these pathways and mechanisms therefore serve and provide targets for therapeutic interventions, which we have employed and exploited over the years. Hopefully, some of these techniques can be used in order to try to combat the effects of atherosclerotic renovascular disease on the kidney as well as on the cardiovascular system in human subjects with this disease.